Hey y'all, Miss O'Leary here. Turns out you can make yourself an animal avatar now on Zoom. So, in light of it being spring, I chose a bunny. So, this bunny is me, and I'm going to be teaching you about replication, cell division, DNA structure, PCR, and mutation today. So, to give a little bit of context, we're going to be talking about cell division um, in light of looking at our oral microbiome lab. So just as a starting point, cell division is making daughter cells. So if you remember, our cell theory states that cells can only come from other cells. And so that is an important part of this is that you get new cells from old cells dividing. This is true for both prokaryotes and eukaryotes, but it works a little bit differently in each of those. For eukaryotes, the parts all have to be replicated first, and that includes things like the mitochondria and all of the other membrane-bound organelles that are inside of the cell. So you can see in the picture on the right, there are all kinds of different organelles that are visible here. This is the actual cell division process, but as you can see when they divide down in late telophase, each of the cells has about the same amount of stuff, and they both look very similar to the first cell that is here in interphase. Prokaryotes also have to replicate all of their parts. That includes the DNA, just like in eukaryotes, but also the cell wall. So the only kind of eukaryotes that have a cell wall that we talk about really are plants, but in prokaryotes, they do have cell wall also that would have to be replicated. So this is a picture of some different bacteria that are dividing through cell division. And so you can see the red parts here are where the cell wall is actually growing so that when the cells divide, and they have complete cell walls by the end. So one of the main things that has to be replicated before cells divide is the DNA. So as you can kind of see here, if we're looking at linear chromosomes, this would be referencing a eukaryote. And so the eukaryote goes from a single chromosome with a centromere. So that's what that circle is at the middle. During S phase of the cell cycle, so this is synthesis or replication, it gets duplicated. Um, Now, I'm a little picky about this. You have to think about these as being two separate chromatids. So they basically look like this first chromosome, but there's two of them stuck together. And so this little kind of X or butterfly shape, as I like to call it, even though we kind of think of that as the stereotypical shape of a chromosome, they actually only look like this after they've been replicated before cell division. So normally in a cell, they do not look like this. They are decondensed, so kind of like the bowl of spaghetti in the nucleus, and they would be in a more linear shape. After mitosis, though, these two copies of the chromosome pull apart. So one thing to note is that this is one chromosome at the beginning. After S phase, this is still considered one chromosome. It's just a replicated chromosome. So I always think of caterpillar, butterfly. So one caterpillar turns into one butterfly, not two. This is kind of where my analogy fails, but think about the two sides or the two wings basically coming apart back into two different caterpillars. And then you do have two chromosomes at the end. But in the intermediate phase with replication, this is still considered one chromosome. So for a human, you have 46 chromosomes that are in this linear state. When they're duplicated, you still only have 46 chromosomes. So you never have more than 46. And then when they divide, you end up with 46 in each cell. In terms of how the chromosome is replicated, that works a little bit differently in different cell types. So this is a picture of bacteria. So you can see that bacterial chromosomes are circular. And then we have something called the origin of replication. So that's just what it sounds like. It's the starting point of replication. So you can see it opens up kind of like a bubble, and then you get new DNA synthesized against each of the original strands of DNA, which then expands and makes two new circles. At the end, one thing that's notable here is that you have to basically unlink the two circular DNA chromosomes, very similar to how in the last picture you have to pull apart the two sister chromatids for a eukaryote. Um, so I'm not going to play through this whole thing, but let me pull up the picture here of the cell cycle. Oops, sorry, a little too far there. Okay, so when you look at this cell cycle, notice that mitosis is actually a pretty small part of it compared to the whole of the cell cycle. Now, I actually would criticize that this is actually misrepresenting how much is actually mitosis, and I would put it as a much smaller part. So interphase is basically everything other than the physical cell division, which is mitosis. Interphase involves having a G1 phase, which is the gap one or growth phase. That phase is basically getting the cell ready for synthesis or DNA replication in the S phase. And then the next phase, the G2 phase, is getting the cell ready for the physical cell division. So those are the main parts of the cell cycle. As you go through and are reviewing this material, you should stop on this slide and play through the video so you can see how this all works. Same thing here. So we have our prokaryotic mitosis. Very similar sort of things are happening here, but you can play through this video and see how cell division looks a little bit differently with prokaryotes. Okay, so up until this point, what we know from both the beginning of this unit and prior units is that cells only come from other cells. Cells divide to create new daughter cells, so that is on par with the first one. All the parts of the cell must be duplicated before cell division so that your new cells are complete and functional. And one of the important parts is the organization of the genetic material and other organelles so that the cell division happens correctly and the new daughter cells are functional. So what we will look at next is DNA replication. So the actual duplication of the genetic material. And to understand that, we need to know what the structure of DNA looks like, how DNA structure and its self-replicating ability can help us in the lab. And this is where we will tie back to our oral microbiome project. And then how does DNA structure replicate in vivo or actually in the cells? If you look over to the right here, this is a little bit more detail about what this duplicated chromosome looks like when you compare it to the structure of DNA. So we all have kind of an idea of DNA being this twisted ladder, double helix. I mentioned histones in my class, at least, as a quaternary structure protein. Histones are octamers. So they are 8 pieces that come together and they act as like the spindles that the DNA is wrapped around. And so the spindle plus the DNA is considered a nucleosome. So you can kind of see us zooming out a little bit here. And then what we get is called supercoiling. So these coils are basically coiled together, and then these are coiled even further. And so this is where we get the condensation of the chromosome. So otherwise, if you laid your DNA out from one cell, it would be very, very, very long from each chromosome. But in this case, it is supercoiled into what fits in the nucleus. I know this is a little old for you all because you're youngins and use your cell phone, but if you think of an old school telephone with the coiled cord, we have those in many of our classrooms. Think of that as being the original coil, but if you ever see them get kind of twisted up on themselves, that would be kind of like a supercoiling thing. Okay, so there's a bunch of information about the structure of DNA on this picture. So there are some details you'll need to go back and review on your own, but just to point out some of the major pieces here. A single piece of DNA is called a nitrogenous, or sorry, a, a single piece of DNA is called a nucleotide. And so this is where you get the deoxyribonucleic acid. Each nucleotide has a nitrogenous base, which is one of these 4 in the corner, adenine, thymine, guanine, or cytosine. And then each one has a sugar, where you can see the sugar phosphate backbone. Each group of sugar and phosphates goes with one base. So in this picture, the first kind of set of 3 things is one nucleotide, and then we add onto them from there. So that would be like one side of the helix or one half of the twisted ladder. The rungs of the ladder, so you can see like the blue and yellow and the red and green matching up here. are held together through hydrogen bonds, which we should know all about. So if you look at the top right here, you should be able to identify some polar bonding here. So NH, for example, is a polar bond. The H's are partially positive. The double bonded oxygens on the other one are partially negative. And so these hydrogen bonds fit together. We'll get back to this in a little bit, but the DNA is where the directions are held for making proteins. And so this is why we will keep talking about DNA for most of the rest of the semester. So just to go through a little bit about the people that were involved in finding out the structure of DNA. So this was in the 1950s. So it may seem like a really long time ago to you all, but in kind of perspective, my parents were alive during this time, and I'm not really that old. So it's compared to a lot of other science and things that we kind of always take for granted, this is actually pretty young. So Watson and Crick were the two main people that most people know related to understanding the structure of DNA. Now, they did not do what most people think about as experimental science, but they did model building. So they basically looked at data and the analyses from a lot of other scientists, and were trying to use that information to construct models of the DNA, which they eventually did. Edward Chargaff, he was one of the people whose data went into Watson and Crick's design. He was a Jewish immigrant during the Nazi rule, so he came to the United States. And he was the one who determined the rules of binding for the base pairs. And then Rosalind Franklin is another one that person who's sort of getting more well known now, but originally wasn't really credited for her part in the discovery of DNA structure, but she did X-ray crystallography. So if you look at that top picture up there, think about X-rays of your bones, how you can see the bone part. This is kind of similar. Think of it like a reverse image. If you look down at the helix from the top, you would see kind of like this plus sign or X shape. And so this picture is part of what they used to figure out that it was a double helix. A lot of people get kind of up in arms about her not winning the Nobel Prize. But actually, one of the earlier things that happened was that Watson and Crick published their paper on the structure of DNA with her picture, this picture up on the right, and did not give her credit. The reason she didn't get the Nobel Prize was partially because of that. She worked in Maurice Wilkins's lab, and so he actually won the Nobel Prize. But also, the Nobel Prize cannot be awarded to more than 3 people or posthumously. And she had already passed away from ovarian cancer at age 37 because of radiation exposure. So even if she was alive, she may not have gotten the Nobel Prize because Maurice Wilkins was basically the person running the lab she worked in. But even if she had really gotten the credit she deserved, because she'd passed away, she wasn't able to get the prize. So just a little bit more about Chargaff here. So in terms of the bases, you can see the ATGC. That's what he kind of figured out, was that those were the base pair rules. And he has a nice quote here: "Science is wonderfully equipped to answer the question how, but it gets terribly confused when you ask the question why." Another picture of Rosalind Franklin here. You can see she also has an interesting quote here: "Science and everyday life cannot and should not be separated." And there are some other people that really still are not very well known among the public for their roles in figuring out the DNA structure. And one of those people was Dr. Marie Maynard Daly. She was the first African American to earn a PhD from Columbia University, which is really impressive. And then also the first African American woman to earn a PhD in chemistry overall. In 1947. So that's really awesome, a great accomplishment. She was actually mentioned in Watson and Crick's Nobel speech because she determined that the ratios of the bases in DNA were not of equal quantities, which led to more of what Chargaff looked at with the base pairing options. And she's also well known for a lot of other things, including finding the connection between cholesterol and high blood pressure. And she also was kind of up in the more administration level and a lot of places that she worked, and had a lot of accomplishments throughout her life. But this contribution was significant for what we're talking about. Another person that I kind of recently learned more about was Dr. Herman Branson. He also contributed a great deal to our understanding He actually helped with protein structures, which we just finished covering. And he very impressively used math to determine the helical shape of DNA. So he was able to go through analyses and figure out that it was either an alpha or a gamma helix in the shape. He worked and published with Linus Pauling. Um, he did not win a Nobel Prize, but Linus Pauling did later win a Nobel Prize. Linus Pauling actually originally suggested a triple helical structure, which they later figured out was not the case. And fun fact, Dr. White got to meet Dr. Branson in college. So that's really cool. You can ask her about him. But like I said, these people are, within a couple generations of you. So this really isn't a long time ago. So the whole molecular biology field is relatively young compared to some of the things that you learn about. Think about what you learn in physics with Newton's laws. Newton was a long, long, long time ago, and work is still important, but as important as DNA structure is, not so long ago. Okay, to get back to the actual DNA structure, you can see the picture of the molecular structure all hooked together with the bonds in this picture. One of the things to note is that it is anti-parallel. So anti meaning opposite, and parallel meaning running basically at equal distances. Now, if you look at this picture, the way to see the anti-parallel nature is to look at the sugars. So you can see that this sugar is a ribose sugar, a deoxyribose sugar. It has 5 carbons, so they're marked in red, 1, 2, 3, 4, 5. This is a little bit silly, but when I was trying to learn how to number them, I would always think of this like a house, and then the 5th one looks like a chimney. So I thought 5 is the fireplace. Other folks can just remember that 1 is connected to the base, if that helps you, and then basically go around from there. Now, why do we number these? This is just for basically a way to orient yourself when you're looking at the DNA, because understanding which direction the bases are running, so which ways the nucleotides are facing, can be important for different processes in the cell. So if you look at it, on the left side, the 5th carbon is facing up on the sugar. On the other side, you can see that the 5th carbon is facing down, and it's the 3rd carbon that's on the top. Another way to notice this is to look at where the oxygens are on the pentose sugar. So on the little house shape, oxygens are facing up on the left and down on the right. The reason for this is that the nucleotides are assembled independently, and flipping them basically one, so you can't see my hand gestures, this little bunny, but if you think about them trying to bond together, the hydrogen bonds will only form if you flip the whole nucleotide on one side upside down. Otherwise, the hydrogen bonds would not bond stably. Okay, so why are we talking about all of this? We are giving this content in order to understand the process of polymerase chain reaction. And we're doing polymerase chain reaction, or PCR, in order to look at your oral microbiome. So PCR is a process of rapidly reproducing billions of copies of segments of DNA. It basically uses the natural structure of DNA to be replicated and takes advantage of that. And then the outcome of PCR can be used for a bunch of different things. It can often be used as a source of DNA for a secondary experiment. So you can actually physically take the DNA and do something else with it. It can be a way to identify a gene or an organism in a solution or an unknown mixture. This is actually how they run COVID tests. This is very similar to what we're doing. And then it can also be used to compare information in between individuals. So this is kind of the traditional CSI DNA fingerprinting type information. And we're going to be doing something similar, except looking at combinations of bacteria in people's microbiomes, oral microbiomes, instead of their actual DNA. So again, to talk about some important people, this is a Nobel Prize winning procedure. It's from 1993, so I was 10 years old then, so this seems really new to me also. This is what we consider a paradigm shift in biology. So paradigms being like a big focus or a change in how things are done. So a change in paradigm is, in this case, a change of how we looked at DNA. So basically being able to rapidly reproduce DNA synthetically in the lab opened up tons of different kinds of experiments and procedures that we could do actually using DNA. Otherwise, we basically didn't have enough quantities of what we were looking at to do very much with it. And PCR was also extremely important as a base for how DNA sequencing worked to do the Human Genome Project, also in the 90s. So just to give you some context of how really smart scientists can also be really interesting and kind of wacky people, Kary Mullis was the person who came up with the procedure for PCR. He actually is known for being a little bit crazy, and he often contributes or attributes his development of PCR to his taking LSD. There's definitely quite a few different stories about him taking LSD and like figuring out PCR while he was taking a bath, that sort of thing. He also, there was a glowing raccoon in one of his papers that he published. He became sort of famous and won the Nobel Prize for this. And so people kind of rubber stamped the things that he wanted to publish. And then it turned out later that some of them were a little bit weird. He also has some interesting, not really scientifically supported ideas, like that HIV hasn't been proven to cause AIDS, which it has, and that global warming and ozone damage were illusions, which also we know is not true. So he's quite a character. Unfortunately, had some of what we would see downsides in terms of his personality, because he was known for womanizing and engaging in some argumentative and sometimes violent behavior with other scientists. And like I said, kind of a goofy guy, but also very brilliant. So just to remind you that scientists are people too, with all of the good and bad that comes along with that. Okay, so I'm going to pause here and then make a second video that goes into the details of PCR.